Like was said, my name is George Ghanim. I'm a new faculty member at Princeton University, and I'm really happy to be able to share some of this work that I did at the very tail end of my postdoc with everyone today. And given the audience, I will spend a little bit of time talking about some of the challenges that we faced during the processing of the cryo-EM data set, as well as maybe some subtleties about some of the AlphaFold predictions that we had done that's in this manuscript. And I would be happy to take any questions during or after. So, right. In our lab, we study the structural mechanisms of LINE-1 and ALU elements. And these are an abundant class of elements that make up a vast majority of our genomes, almost a third. And you should really reflect on that because every cell in your body has a genome, nearly every cell, and a third of that is derived from these elements. So they're massive players. But importantly, they're the only known active retrotransposable elements, and they move by a process called retrotransposition. They retrotranspose. What does that mean? That means that they can copy themselves from one location into the genome and then insert themselves into another. And you'll immediately recognize that that sort of activity can be mutagenic. And it's true, retrotransposition causes DNA breaks, it disrupts genes, it can drive very sophisticated oncogenic rearrangements, and it's recently even emerged as a hallmark of certain malignancies. So these are things that live within our genome, they can move, and they can cause disease. Okay, so how does this all work? We'll focus on the LINE-1 element for this talk, and the LINE-1 element contains two open reading frames, ORF1, which is a nucleic acid chaperone, and then ORF2, or ORF2P, which is an endonuclease and reverse transcriptase. It has endonuclease and reverse transcriptase activities. And retrotransposition begins with transcription of the LINE-1 mRNA, and then in cytoplasm, ORF1 and ORF2P will assemble with their own encoding mRNA. Not other LINE-1 mRNAs, not other cellular RNAs, but their encoding mRNA. And this is called cis-preference. You don't need to remember it, but it's just an unusual feature. Okay. Together, ORF1P and ORF2P with the LINE-1 mRNA will form a ribonucleoprotein complex, or an RNP. And after nuclear entry, ORF2P will nick the bottom strand, the target DNA on a bottom strand at this consensus sequence, producing a 3' end, which is used to prime reverse transcription of the LINE-1 mRNA. So I'll just repeat this. ORF2P nicks a target DNA, that produces a 3' hydroxyl, and then that 3' hydroxyl primes reverse transcription of the LINE-1 mRNA. This is called target primed reverse transcription, or TPRT for short. Now, I'm leaving out a lot of details here, but this will eventually lead to a new LINE-1 insertion. Okay. Now, many aspects of this mechanism and this life cycle of LINE-1 mobility are poorly understood, and I'll highlight two examples. The first is the mechanism of TPRT is unclear. It's not really understood how DNA is recognized or how it's organized within this complex. Next is, it was unclear if ORF2P completes this insertion process. A model was proposed that cellular factors complete this insertion process. So completing this insertion process would require removing this LINE-1 mRNA, nicking of the top strand, and then using that top strand to prime second strand synthesis of this cDNA, generating this double-stranded insertion. And again, it was unclear if ORF2P nicks the top strand or if ORF2P can synthesize the second strand. So to answer some of these questions, we sought to solve the structure of the TPRT complex. And this is the complex where ORF2P is initiating that reverse transcription reaction into the target DNA. So to start, we purified ORF2P. And for anybody that's worked with transposable elements, they know how difficult this be. And it was exceptionally difficult. And I'll leave all the details here out. And I'll just say that we were able to purify ORF2P. And not only that, we were able to purify ORF2P to a high TPRT activity. And we reconstituted this TPRT activity using a fluorescent target DNA. And importantly, this fluorescent target DNA, this target, was derived from the factor VIII gene from a de novo insertion event that's in a patient that's known to cause a disease. Now, in these assays, we give ORF2P an RNA, we give it a target DNA, and then we look for it to reverse transcribe the RNA into the target DNA, and that would result in these slower mobility species up here. And when we do this with an unnicked target DNA, with that consensus sequence that ORF2P likes to target, we can barely see activity. So we instead switched to a target DNA that was pre-nicked on the bottom strand. Recall that this target sequence is derived from a de novo insertion event that had caused disease in a particular patient. And by introducing this nick, we're mimicking an intermediate of the retrotransposition reaction, the intermediate that occurs after bottom strand nicking. And you can see that by using this pre-nicked substrate, it stimulates TP activity very high. But importantly, if we mutate this TTAAA sequence, this consensus sequence, to something else, we see that the stimulation is lost, despite the bottom strand still being nicked. So there's something about this sequence, this consensus motif that the ORF2P is specific to. And it's probably annealing with the poly A sequence is why this is required. Okay, so using this pre-nicked substrate, we were able to reconstitute this TPRT complex. And again, this is the complex where ORF2P is copying its mRNA into the target DNA. And we were able to solve not just one, but 4 structures by cryo-EM up to 2.3 angstrom resolution. And these structures represent different states, different conformational states of the enzyme. And we'll go into them a bit later, but for now, we'll focus on the consensus reconstruction, which is on the left. And if we pull up a schematic on the right, just to help orient you, you could see that in the spinning movie, ORF2P, which is colored in gray here, is really forming a basket around the nucleic acids. You can see the 2 parts of the target DNA, say here, so one is here, and the other is in the active site, and it's annealed with the RNA, as we would expect. And we find that ORF2P can be divided into 6 domains, and these domains come from work that was done previously as well. So we start with the endonuclease domain, but we don't see this endonuclease domain, and it's because it was flopping about, and you can see it flopping about in our 2D class averages. You could see it here and here. So in our consensus reconstruction, we don't see it. Next is what is called the EN linker domain, which we'll just refer to as linker. This is followed by the N-terminal extension domain, or NTE. And collectively, these have also been referred to as the tower domain. This is followed by the RT domain, or reverse transcriptase domain, the thumb subdomain, the wrist domain, and then finally what is called the CTD, or C-terminal domain. And within the CTD, we see clear coordination of a zinc ion by a zinc finger. Now, for the rest of this talk, it's only really important to remember the CTD and the RT, and then when it's appropriate, I'll bring back the endonuclease domain and the wrist. But for now, just the CTD and the RT. And we were able to catch the reverse transcriptase domain in different conformations. And we'll take a look at some of these structures, these other conformational states that we solved. So we'll focus in on the RT. And just to remind everybody, structurally, reverse transcriptases and DNA polymerases resemble this hand architecture. And they contain 3 subdomains. So the first is the thumb, the second is the palm, and the third are the finger subdomain. And each of these subdomains play a particular role in handling the nucleic acids during the catalytic cycle of reverse transcription. But for now, we'll just focus on the fingers. And the fingers open and close during catalysis. And the idea is that they open and close over the active site. And after proper base pairing is formed, that drives the addition reaction forward that catalyzes nucleotide addition, the closing of the fingers. And that's what we see in our data. We find that there are these two conformations. And I'll take a moment just to talk about the processing. In the consensus reconstruction, we could see that this region was very poorly resolved. And we tried several methods. But what seemed to be the best was to do focused 3D classification in CryoSpark, actually. So the data is primarily processed in LINE-1, and we move it into CryoSpark for this to find these particular states. And this strategy has proved relatively successful, especially with the force hard classification feature. And by testing a lot of things, we were able to discretely pull out these two states. And these two states represent the open fingers and the closed fingers. Now we'll take a look at the fingers in the open conformation. And here, the fingers are opened and positioned upward. If we zoom in, and you can see that the catalytic site is empty. So additionally, the catalytic residues, which are these acidic residues here, are not properly positioned for catalysis. Okay, but in the second conformation, these regions close over an incoming nucleotide, and this closed state catalyzes nucleotide addition. And again, we were able to find that state in our data set. So in this closed conformation, you can see that now there is a nucleotide in the active site, and it's making important contacts with particularly the fingers as well as the palm. And when we zoom in, you can see that now these important residues are positioned for catalysis, primarily these acidic residues, which coordinate this magnesium ion. Okay. So when we overlay the two structures, you can see the type of conformational changes that we were able to distill from our data sets. Now we'll switch and we'll take a closer look at the nucleic acids in the structure. And to me, this is the most fascinating part, and it's really the most unexpected part. We find that ORF2P is extensively remodeling the target DNA. And this was completely unexpected because recall, the target DNA was presumably just nicked on the bottom strand. It's designed to mimic intermediate after nicking by the endonuclease. So presumably the target DNA is just nicked on the bottom strand, but within our structure, we find that ORF2P is effectively breaking the target DNA into these two parts. And for clarity, we'll refer to the first part as the first primer region, because it's priming first strand cDNA synthesis. And then the second region of the target DNA as the second primer region, because presumably it would prime second strand synthesis. We can zoom in on the first primer region, and we can rotate this towards us and cut off the top to get a clear view of the active site and the target DNA. And we can see here that it contains the RNA, which is colored, annealed to the target DNA. And the target DNA here, the first primer region, is priming cDNA synthesis in the RT active site. And this was completely expected. Okay, but what was not expected is what's happening at the second primer region. So we can zoom in, and we find at the second primer region, it appears to be pinned at one end by the CTD. And this is just a rotated view of this image here. I hope you can see my mouse. We find that it's pinned at one end by the CTD, and that the CTD is wedging itself between the two strands of the target DNA, as if it was melting or unzipping the target DNA. So this was evident to us during data processing, where we see these different lengths of the target DNA and at the second primer region. And I'll just play this again. And what these different lengths correspond to is basically different states of melting of this second primer region. Okay. The unzipped bottom strand, the melted bottom strand here is disordered, and we don't see it in our reconstructions, although we see maybe one additional nucleotide in some of our in the data set. But the top strand is held tightly by ORF2P. And we'll take a look at that here. And here again, we're rotating the structure to take a look at the top strand. And it's held in a way with the bases facing the solvent. It's also ideal B-form DNA geometry. This region over here. So the bases are exposed to the solvent, and the enzyme is basically holding it from the phosphates. And as far as we could tell, not really making any base-specific contacts, although it was very difficult to assume the identity of the bases here. It's a little bit unusual for an enzyme to have the nucleic acids exposed to the solvent and the bases exposed to the solvent. And we're currently working out what that means. Okay, so we'll move on. And one of the things you may have noticed is that the two DNAs are separated, almost as if the top strand was nicked. And again, this is a little bit unusual because presumably we're starting with a target DNA that's just nicked on the bottom strand. But despite a lot of the processing and looking at our structure very carefully, we couldn't find a way to connect the two target DNA regions. So we asked, is the top strand being nicked? And we tested this using our TPRT assays. But this time, we're doubly fluorescently labeling the target DNA. So Cy5 on the bottom strand and FAM on the top strand. But it allows us to track TPRT or cDNA synthesis on the bottom strand, and then any nicking or potentially second strand synthesis that would happen on the top strand. So when we look at the bottom strand in this time course experiment, we see that there is an accumulation of this TPRT signal, this cDNA synthesis. This is expected. But then when we switched and looked at the top strand, we see that there is this corresponding accumulation of these faster mobility species. And these are nicked top strand products. Importantly, if we mutate the endonuclease domain, we see that this has no effect on TPRT. It slightly diminishes TPRT activity, but it's not really significant, I think. But it completely eliminates this top strand nicking. So it's clear that the endonuclease domain is nicking of ORF2P is nicking the top strand of the target DNA. We went ahead and mapped these cleavage sites by Sanger sequencing, and I'll just take a moment to remind you that the target DNA is derived from a patient, and here is the insertion consensus site of that target DNA. And we're nicking the bottom strand here. And in that patient, presumably, the top strand is nicked here. So we went ahead and mapped these cleavage sites by Sanger sequencing. And you can see that the first cleavage site is here, the second cleavage site is here or here, and the third is here. So they're different from what was observed in vivo in the patient. So overall, we find that ORF2P is nicking the top strand, and it seems very likely that it's carrying out second strand synthesis. It seems to be positioning the nucleic acid, particularly of the second primer region, such that it can rapidly switch to the second part of the reaction where it does second strand synthesis. Okay. We were also able to resolve that endonuclease domain. Now, remember in the beginning of the talk, I had mentioned that this endonuclease domain was very flexible. And here's the 2D class average where we see this endonuclease domain flopping about. And in a subset of the particles in the data set, the endonuclease domain seemed to adopt a more stable conformation. And again, we'll take a moment just to look at the data processing because this was quite challenging, actually. And I don't think I would have been able to do it without the help of Johannes, who was a postdoc in Shor Sheraz's group at the LMB. So the way we find this endonuclease domain is, again, bringing the data set from RELION into CryoSpark and doing focused 3D classification. And eventually, after trying many things, we found that in some subsets, we see this bump, right, which we presumed was the endonuclease domain. We bring this data back to RELION, and we do more focused 3D classification, and we're eventually able to find resolve this flexible domain. So one of the challenges here, I had tried many methods at the time, and one of the challenges are these two long helices. So the endonuclease domain is flopping about, and it seems to be contacting these two helices, but the rigidity of the helices seem to dominate the alignments in a lot of the strategies I was trying. Normally, I think if these alpha helices were not there, some of these easier strategies would have worked. And I think I would have been able to resolve the endonuclease domain to higher resolution. But because of these two rigid helices, I say rigid, they weren't even that rigid. They would flex back and forth with the endonuclease domain. It complicated the data processing. And one of the things we had done was take this into multi-body refinement, where we fix that endonuclease domain and look at the principal components of the rest of the protein. And again, by realigning back to the bottom, we can see that this endonuclease domain is basically flopping in every possible orientation that it can, while still maintaining contact with these flexible, these two long helices in the back. Okay, so we can resolve the endonuclease domain, and we find that in a subset of the particles, it is positioned up against these two long helices of the linker domain. And it's making contacts here, here, and here. And this is important because previous studies had carried out scanning mutagenesis experiments of ORF2P and ORF1P. And if we take that data, that scanning mutagenesis data, and map it onto our structure, we could see that the regions that are interacting with the endonuclease domain here, here, and here, if you mutate them to alanine in this experiment, you lose retrotransposition activity. Whereas neighboring regions don't have this issue, like this other alpha helix in the back. Mutations there don't seem to have as profound as an effect on retrotransposition. So we think that this positioning of the endonuclease domain on these helices represents almost like a default configuration of the domain. And it explains a feature of retrotransposition called target site duplications. And I didn't have the time to really get into this, but I'll explain it now. New line-1 insertions are flanked by a direct duplication of the target site. You could see the new line-1 insertion here, and it's flanked by these TSDs, these target site duplications. Okay, and this arises because of the staggered nick of the bottom strand relative to the top strand. So we nick the bottom strand here, but if you nick the top strand here, you produce a duplication of one base pair on each side of the new insertion. If you nick the top strand here, you get two base pairs. If you nick the top strand here, you get three base pair duplication on each side. Okay, so on and so forth. You get the idea. But for most of the transposases, particularly DNA transposases, they tend to produce a very defined target site duplication. But for line-1, the lengths vary. The lengths vary from 7 to 20 base pairs. The average is about 16. There's some that are much larger, some that are much smaller, but it's about 7 to 20. And we didn't really understand why the target site duplication should vary at all. And we think that our results help explain this, because modeling of target DNA that's nicked on the bottom strand into this structure shows that in this configuration, the site of top strand nicking is positioned about 20 nucleotides away. And again, we think that this positioning of the endonuclease domain represents some sort of default configuration for top strand nicking. But as you saw, ORF2p sliding, or more importantly, melting of this target DNA would draw this bottom strand nick towards the endonuclease domain. And that would result in bringing the two nick sites closer together and then resulting in target site duplications that are less than 20 nucleotides. So we think that where we find the endonuclease domain explains a feature of retrotransposition. What it doesn't explain is the nucleic acid specificity here. And many of the interactions that we saw in our reconstruction between ORF2p, the protein, and the target DNA, as well as the RNA, was made through the phosphate backbone and did not appear to be base specific. And this is really, it was a little bit unexpected because retrotransposition has several nucleic acid specificities. And I'll just give one example. The poly-A sequence is essential for retrotransposition. LINE-1, the ORF2p protein, is known to reverse transcribe itself. Again, this is the cis-preference I had mentioned. And it's not contingent on a sequence within the LINE-1 mRNA or some sort of structure, but just the poly-A itself. So it has some sort of specificity, but it doesn't seem to be dictated by base specificity, although it does have the specificity for its poly-A sequence. Okay, so despite this need for a poly-A sequence on its mRNA, we don't see any base-specific interactions with adenosine in our structure. Now, it's known that many cellular proteins associate with LINE-1 and are required or facilitate the retrotransposition process. So it raised the possibility for us that maybe some of these proteins are playing that role, where they are guiding LINE-1 or dictating the specificity at particular stages. So we did what any structural biologist does nowadays, and we predicted AlphaFold structure. We predicted AlphaFold structures and between ORF2p and known interactors. And of the handful that we predicted, two came up as high confidence and seemed to be very interesting. And the first is PCNA. So PCNA is the clamp during DNA replication, and it acts as a processivity factor for the replicative polymerases to vastly increase their processivity. PCNA is also important for other aspects of DNA replication, such as resolution of Okazaki fragments, as well as in DNA repair. And we find that there is a convincing prediction between ORF2p and PCNA, and it was previously known that they would interact, but not exactly how. And here's a rotated view, and we find that ORF2p or PCNA is binding a novel site on ORF2p, and it's occurring through this wrist domain of ORF2p. So we named this the PUB, PCNA unusual binding motif, PUB for short. And we see that ORF2p is engaging this hydrophobic pocket of PCNA through a helix rather than the typical short peptide, which many proteins use to interact with PCNA. And here's a close-up view of this interaction on the right. And so there's several pieces of evidence that support and validate this prediction. The first is if we take the scanning mutagenesis data that I had mentioned previously, and again, map this onto our structure, we see that point mutations in this PUB region, in this PUB helix, substantially affect retrotransposition. They basically kill it. Whereas again, substitutions in neighboring regions like this loop and helix back here don't seem to have as profound as an effect. So there is also a very high conservation of these residues that interact across very diverse LINE-1 elements. And this is shown here on the bottom. And then finally, we went ahead and did a little bit of biochemistry. And we find that the pub mutants, particularly the ones that are interacting, making these interactions, this tryptophan and this isoleucine, mutating this to alanine diminishes the ability of ORF2p to associate with PCNA in pull-down experiments. And what's quite nice about this is that the effect seems to scale with the hydrophobicity of the residue that's mutated. Okay, so we also, in our predictions, found that poly-A binding protein seems to bind near the RNA entry channel of ORF2p. And this is the AlphaFold prediction. And poly-A binding protein, or PABPC, regulates really many aspects of mRNA biology. Its stability, its translation, so on and so forth. And it belongs, poly-A BPC1 belongs to a larger family of poly-A binding proteins. I'm not an expert in poly-A binding proteins, but I think it may be the most abundant protein in the cytoplasm. And previous studies showed that poly-A binding protein is part of the LINE-1 complex and that it promotes efficient retrotransposition. And we find that poly-A binding protein, or the region that's shown here, is predicted to interact with the long linker region, these long helices. And it is right near the entry and exit channel, the RNA entry and exit channel of ORF2p. So we name this element pi, or the poly-A binding protein interacting an essential element. And this is almost like a homage to this work, which was done in the United Kingdom. And if you've spent any time in the United Kingdom, you know how important pies and pubs are. So it seemed only appropriate to name these motifs that. And when we zoom in, we could see that this interaction is primarily made with the RNA recognition motif of poly-A binding protein, as well as this region of ORF2p. And again, this is supported by 3 pieces of evidence. The first is if we take this scanning mutagenesis data and map it onto the structure, we could see that substitutions at these regions that are predicted to interact strongly diminish retrotransposition, whereas again, substitutions at neighboring regions, or this helix back here, which I'm not showing in this figure, don't seem to have as strong as an effect. Next, the residues are highly conserved across diverse line-1 elements from other organisms. So here they are highlighted with these stars, and you can see that there's relatively strong conservation, particularly in this region over here. Okay. And third, ORF2 mutants, poly-A binding protein does not co-localize with ORF2p mutants that are mutated in this, this pi motif. So here you can see in these IF images, we have, we're staining for poly-A binding protein as well as ORF2p, and you can see that they co-localize from these puncta. For whatever reason, ORF2p seems to form puncta when it's expressed alone. Fine. And that it co-localizes with poly-A binding protein. And here's a few examples highlighted here, here, as well as here. But when we make point mutations in ORF2p that disrupt this predicted interaction interface, we see now that there is no longer any co-localization. Again, here is poly-A binding protein, this diffuse signal. We see some ORF2p puncta, but again, it does not seem to be co-localizing to as strong as an extent as the wild type, or at all. So this co-localization is lost when we overexpress ORF2p mutants carrying mutations at this interaction site. Now, overall, these results are very appealing to us because it may explain something about LINE-1 that I had mentioned in the beginning called cis-preference. And recall that ORF2p preferentially associates with its own encoding mRNA, not other LINE-1 mRNAs, not other cellular RNAs. And this is cis-preference. And it's known that cis-preference requires a poly-A tract, and it's thought to occur co-translationally, but it was still unclear how ORF2p selects its own mRNA in a sea of mRNAs just by the poly-A. And we think that this is happening, that the interaction of ORF2P with poly-A binding protein may be the basis of the line-1 cis-preference. And this pi motif, this interaction site, is located very close to the N-terminus of ORF2P. So during translation of ORF2P, poly-A binding to this pi site could potentially establish cis-preference and could potentially position the RNA for co-folding of the reverse transcriptase domain on the RNA even before the reverse transcriptase has even been synthesized. So we think that it might explain this feature called cis-preference. Okay. It's really an attractive model because not only does it explain cis-preference, but it would act to bring ORF2P to the very end of its mRNA. It's known that poly-A binding protein coats the poly-A track, and each protomer occludes RRM of the previous protomer, such that only RRM1 of the last protomer is exposed for potential interaction. So it's an attractive way, it's an attractive model for how ORF2P finds the very end of its mRNA. Okay, so overall, we reconstituted the line-1 TPRT complex, and our reconstructions show a sophisticated and complex arrangement of nucleic acids and proteins. The structure rationalizes several features of retrotransposition, namely the varying lengths of the target site duplications. We find that the target DNA is melted, the endonuclease domain is flexible, and that, importantly, the endonuclease domain is nicking the top strand. So it seems very likely that ORF2P is or will complete that retrotransposition process. And then finally, structure predictions give insights into retrotransposition. It's possible that ORF2P is acting to exploit PCNA to find a target DNA rather than using PCNA as a processivity factor, which is what the replicative polymerases do. And then the interaction with poly-A binding protein may explain this cis-preference that line-1 has for its own encoding mRNA. So, it's shown here. Yes. So with that, I'd like to thank the members of my postdoctoral lab. Like I had said, this was done in the last few months of my postdoc when I was in Kelly Nguyen's group. So big thanks to Kelly, my mentor, as well as the EM facility and all of our friends at the LMB. Like was mentioned, I just started my own group at Princeton where we'll be studying, really taking a deep dive into the structural mechanisms of line-1 and a related element called ALU. And this is a project that I started in the UK, but I will be continuing here. And if anyone is interested in joining our team, studying retrotransposons, doing some structural biology, I'd be happy to hear from you. So please reach out. And with that, I will thank you, and I would be happy to take any questions.
Okay, there's one question in the chat for you so far. Regarding the poly-A structure, or the poly-A sequence, I guess, determining sequence specificity versus other regions or shape complementarity, I guess, can you comment more on kind of the sequence specificity and whether that's sequence-based, shape-based, etc.?
That's a good point. Yeah, because it is known that the poly-A tract adopts A-form structure. I don't know. It doesn't seem to be that way in our reconstructions. The particular state that we caught the enzyme in does not seem to have a state where it would interact with an A-form stretch of single-stranded RNA. We catch it basically annealed with the target DNA already. That's an interesting point, and I'm curious to experimentally, what would happen if disrupting the structure, such as with G's, would have an effect. I think it would. Yeah, I don't know. I would not be surprised if structure did play a role in addition to uh, poly A binding protein.
I have a general question on kind of what you're thinking about for those exposed bases and kind of your first structure you talked about. So there was actually a Cas16 target that was this very cute little uracil DNA degrading protein that had just single-stranded DNA attached to it and also had all the bases exposed and just the backbone interacting, which is a super interesting, kind of strange way, something that normally we wouldn't predict happening. So I'm wondering what you think that means generally for specificity, or like, is it exposing it for a different reason, maybe another RBP coming in and recognizing that sequence, or what are your, can you elaborate a little bit more on what your thoughts are on that signal?
Yeah, I'm glad you brought it up. I am very keen to take a look at those structures now and learn more about these exposed bases. It was really surprising to us, and even though we had done many AlphaFold predictions before we had a reconstruction, none of them seemed to agree with the DNA. The predictions did not support what we saw in our reconstructions for that particular region. What we think is happening is that that region may be priming second strand synthesis. So retrotransposition is basically four steps: nicking, reverse transcription, nicking, and then reverse transcription, but on the top strand. So the bases may be exposed and may be poised to prime with the second primer region of the target of the cDNA. So how we're thinking about it is that the enzyme may be holding that region of the DNA exposed such that it is poised to prime and initiate that second set of reactions that completes the line-1 insertion. I hope that was clear. That's what we think. It's a little bit challenging to experimentally demonstrate, but this is something we're thinking about and thinking about ways of how to show this. Should I let you read the questions, or should I? I think you're muted too.
Or if any of the people asking questions, you're also welcome to unmute yourself and ask as well. Give it. Oh. Okay. They don't want to. I'll start with the first question. For the RT and CTD, do you have any insights how they compare to telomerase RT domain? Kind of looking across your work here.
I do. Here we go. So for the CTD, and you'll have to forgive this slide, this was made when we used to call it the ZZD, but the reviewer wouldn't let us have that, which is okay. But the CTD or ZZD doesn't seem to have any similarities with telomerase per se, but we do find some analogous regions with what is the CTE of telomerase, or the thumb. And I didn't mention it here, but retrotransposons are a little bit unusual in the reverse transcriptase. They're not like HIV reverse transcriptase. They have additional regions that help them do the things that retrotransposons do that retroviral integrases, reverse transcriptases do not. And one of those regions is called the NTE, and it's important for template jumping or template switching. And we do seem to find an analogous region in the CTE of telomerase. This kind of makes sense because telomerase is thought to have evolved from a retro element. So these parallels are quite neat. For the CTD, there really isn't one that we could find that was very apparent. There are a lot of parallels with other retrotransposable element machinery, but not with telomerase.
Right. And then we have a question about off-target effects. So line-1 machinery can sometimes capture off-target RNAs instead of its own mRNA, resulting in pseudogenes. So how does your hypothesis explain off-target effects?
Totally. And this is a really great question. And I'll give a short answer and then I'll give more context. We think that it's just a preference. It's true that line-1 has a preference for its own element, but it will mobilize other things like mRNAs, and that will result in a pseudogene. Another famous example is the ALU element. The ALU is just a structured RNA. It doesn't encode any proteins as far as we know. And it's able to break the cis-preference of line-1, and then it steals ORF2p and then uses ORF2p for its own retrotransposition. So it is a little bit unsatisfying that, so the model is satisfying in that it explains how it's able to find its poly A in a sea of translating poly A's, but it's a little bit unsatisfying in that it doesn't necessarily explain cis preference perfectly, in the fact that by mistake, line-1 will mobilize other things. It may just be a probabilistic thing where there's a very high probability that it will interact with its own poly A binding protein, but every so often there is a poly A binding protein of a different mRNA transcript nearby, and it just accidentally interacts and hops on there. That's really the best way we can reconcile that. I don't know if there's an active mechanism other than it being some sort of chance thing.